Good afternoon, judges. My name is Dr. Shumik Biswas, and I'm the founder and CEO of Tenomix. As we all know, cancer is a terrible disease that personally affects each and every one of us in some way. In 2020, almost 27,000 Canadians were diagnosed with colorectal cancer. And shockingly, this ends up equaling to 73 Canadians being diagnosed every day. Now, when we look at the colon cancer staging process, part of it involves pathology staff manually searching through surgically removed colon cancer tissues for lymph nodes, which can take anywhere from 1 hour to over 2 hours for larger samples. Now imagine yourself in the shoes of pathology staff who are doing this tedious search process by hand, trying to find small lymph nodes that are the same size of a pencil tip. Does that sound reliable to you? Through customer discovery, we confirmed that this process is indeed tedious, expensive, and unreliable. And what's shocking is that lymph nodes can be missed in 30 to 60% of North American cases, which can lead to inaccurate cancer staging and improper treatment for patients. And pathology staff desperately need a solution that can help decrease the labor, reduce costs, and reliably locate lymph nodes. After hearing their pain points, at Tenomix, we've developed the Lymphinator, which is a novel benchtop robotic scanning device that automates the lymph node search process. With our patent pending technology, we're simply changing the tedious search process, which is the key bottleneck to accurate cancer staging. And by integrating early feedback from pathology staff, our device offers several benefits, which include seamless workflow integration, better lymph node detection, set and forget operation, and reduced costs for pathology labs. And this is how the Lymphinator works. Pathology staff only need to place a sample in our device, press the start button, and then our device will do the rest of the work, where ultrasound imaging and software are used to identify lymph nodes that then guide a robotic arm that marks lymph node locations for easy extraction by pathology staff. From our early experiments with our alpha prototype, we've been able to locate small lymph nodes that are difficult to find by hand. And the need for our device is also timely, because with increasing colon cancer cases, ongoing pressures faced by pathology labs, and the growing demand for lab automation, it's the perfect time for adoption. In terms of current competition, there are no direct automated competitors. However, there are alternative methods available, but they have significant drawbacks. By validating our device in the market, our device offers significant advantages compared to existing solutions, which include less labor intensity, cost savings, accurate lymph node identification, and increased throughput for pathology labs. Following a proven business model for medical devices, we'll use a B2B model involving sales to hospitals, which include hardware sales and a recurring revenue model with device consumables and software subscription. Based on comparable tech in the market and procurement thresholds, the sale price of our device is $100,000, and hospitals can see an attractive ROI of less than 1 year. For our go-to-market plan, since we've already engaged with several key opinion leaders, we'll first target large North American academic hospitals that have pathology teaching programs. These trainees will be trained with their device, and they will then be our advocates after their programs, which open the doors to non-academic and private pathology labs. The labs in Canada will be our initial validation market, and we're also actively pursuing several great leads in the US. Now, once expansion takes place into the US and global labs, we'd be looking at a massive $559 million opportunity each year. And I have to say that none of this work would be possible without my amazing team. With expertise in pathology, engineering, business, and medicine, my team and I have raised over $370,000 in non-dilutive funding, and we've already started clinical testing with human tissues, all within 8 months. We also have expert mentors in pathology, software, and business guiding us. Going forward, we'll continue with software development. And in terms of IP, we filed a provisional patent last July. So we'll convert this to a PCT this July to capture a larger market share by filing in countries with a high rate of colon cancer. From there, we'll then finish our beta prototype for a clinical pilot study early next year. And as we work with our consultants, we'll move towards a Class 2 regulatory approval and then plan for production and sales in 2024. Based on our 5-year financial projections, in the first 2 years, we'll focus on R&D activities and will require an initial investment of $1.2 million to move to regulatory approvals. Sales are expected to start by year 3, and we anticipate a 300% annual growth rate after that year. In terms of the Synapse Award, the funds will be used for prototype refinement, IP, software development, and for a clinical pilot study in Hamilton. This funding will help us reach 3 significant milestones, which you can see on the right. We also plan to use the in-kind services for legal support and further networking to grow the business. Having lost my aunt to improper colon cancer staging at a young age, this work is a tribute to her and millions of others who have lost their lives fighting cancer. My team and I are dedicated to help patients get the best quality cancer care. And here at Tenomix, we're ready to fight for cancer patients, one lymph node at a time. Thank you.
Great job, Samik. Thank you so much for that. You've given us lots of areas where we can ask questions, and what we'll do is we'll go around the judges one at a time and come back in a circle and see how many questions we can cover off. So maybe first of all, I'll hand over to Lou for the first question.
Thank you, Jane, and great presentation, Samik. Thanks. Just curious, you're starting with colon cancer. What other tumor types you think can be used for this device?
Lou, it's a fantastic question. So that's sort of the interesting application potential for our device, because for a lot of other cancer tissues, for example, head and neck cancer, breast cancer, lung cancer, gastric or stomach cancer, bladder cancer, they all require some form of lymph node dissection. So we definitely see our device being applicable to these other different cancer tissue types. The other thing to keep in mind is that my team and I, we've looked at other market segments as well. So I'll jump here. We also noticed that animal tissues also require lymph node dissection. So we have the veterinary markets available. And you can sort of see here that we have some of these other cancer tissue types that our device could be applicable to.
Thank you.
Very welcome.
And moving to Alex, do you have a question?
Yes. First of all, again, I'll echo the comments that Lou made, that very nice presentation and a good solution to a real problem. One of the questions I have is whether or not you are planning in your IP strategy to look at somebody who might try to piggyback along your hardware sales and try to develop a competing software platform, especially since you're using a subscription model, where they might try to eat into your recurring revenue by providing an alternate software platform that can execute on the hardware.
Mm-hmm. Alex, that's a fantastic question. And I have to say that in terms of our IP strategy, we are definitely looking to work with one of the legal firms for IP counsel for sure. But there's a few things to keep in mind. Now, pathology is pretty unique from that standpoint, as you were mentioning. So there's a few factors that we have to consider. One is brand loyalty. So we know that with traditional, with a lot of the processes in pathology, a lot of the workflow is pretty traditional and manual. So this sort of presents a natural barrier for new entrants, because with us, what we're trying to do is we recognize this as a potential barrier. But for a go-to-market strategy, we're already partnering with teaching hospitals. And by working with them early on, we are working with the next generation pathology staff, and that we're hoping that would foster some sort of brand loyalty with our device. And that's sort of one of the aspects. We're also sort of incorporating the feedback from pathology staff into this as well. Now, maybe to answer your question in more detail, for other competitors. So if they were to replicate, let's say, the hardware or software aspect of our device, if they created a knockoff, there's an aspect known as vertical integration. And the thing to keep in mind is these competitor firms would have to have control over the supply and distribution of an open architecture USB ultrasound probe company. So to mitigate this barrier, we're actually working with an ultrasound OEM who is a market leader in open architecture USB ultrasound imaging. And they're the leading manufacturer of this ultrasound imaging solution. So for us, because we're working with them very closely, we're hoping to have an exclusive distribution arrangement established with this ultrasound OEM. So that would tie up a lot of the distribution channels for a lot of the competitors. And the few other things to keep in mind too is this network effect. So in pathology, it's a very close-knit community, and it's actually quite difficult for new competitors to enter the space without a critical mass of users. And especially this is more apparent when pathology staff may be more reluctant to use a competitive tool that their peers aren't using. So from the end of Tenomix, we're really hoping to leverage existing pathology networks within our team, engage the KOLs, and also present at key pathology conferences. I hope that answered your question.
Thank you.
Great, thank you. And I have some sort of very basic questions about the technology itself and what you're actually finding. So when you go searching for the lymph nodes in this tissue, the reason for doing it is to help with staging the cancer, correct?
Sort of, Dr. Hamilton. So we're locating the lymph nodes in the tissues, but yes, you are correct that these lymph nodes are ultimately submitted for more tissue staining. So H&E staining, for example.
Right. So potentially you can be better than standard of care because you could find more. Okay, so here's my question. It's kind of philosophical almost. If you're better than standard of care, all the data collected to date is with standard of care. So does it turn up the whole staging on its end? Like, is your staging going to be different from the next hospital that does it the old way?
Dr. Hamilton, that's a fantastic question. And that's something that we're actively building on our study design. So right now, you're 100% right. We're in the sense that if they do miss finding the small lymph nodes, typically it's the lymph nodes between 1 to 5 millimeters that are difficult to find by hand. Theoretically, if they miss these lymph nodes, then the patient's cancer can't be staged accurately. So with our clinical studies that we're planning to develop with Hamilton Health Sciences, we would do a head-to-head comparison study with our device and in terms of the standard of care right now, which is its manual dissection.
Right. So but you could potentially find patients in worse shape than otherwise.
We hope so. Yes, we hope to.
Right. Okay. I think we've time for another round. So Lou, do you have another question? And you're on mute.
Sorry, yes. Just again, just a follow up on that. I guess now with this device, you potentially have more information about the tissue than maybe a dissection has. So is there more information provided from the device would give you a better information to triage patients and to design treatment plans with the additional information?
Lou, that's another wonderful question. And again, this is something that we're actively working with pathology end users. And just to give you some context, we actually had a conversation with a colorectal surgeon today. And we asked them, hey, from this pathology report, what do you feel is missing? And they mentioned that, when these lymph nodes are being assessed by pathology staff, usually there's between anywhere from 10 to 50 lymph nodes being extracted. And the positive lymph nodes, the number of lymph nodes are being identified. But on the report, it's not really clear the position or location or orientation of those lymph nodes in comparison to the tumor. So with our device, we hope that it can help with the pathology workflow and help them better document information such as the positional status or location status of these lymph nodes that could be ultimately used for the standard of care for the patients. And the colorectal surgeon would benefit from this information. Same with the medical oncologist and radiation oncologist. So there's definitely potential to add additional information, as you mentioned, but that's something that we're actively working with our pathology staff.
Thank you.
I'm just going to jump in with a follow-up on there. Have you engaged data scientists? Is there an AI element here as well to exploit?
Dr. Hamilton, for sure. That's a great question. So the AI element, we're approaching the software from a rules-based image processing detection method. So basically to better clarify what I mean by this is that we have sort of established certain set of rules for identifying these lymph nodes. So we know exactly what our algorithm is finding out. Down the road, a more AI or machine learned method could definitely be investigated. But from the standpoint of regulatory, it would be something that a rules-based detection method would provide more clarity on how our algorithm is actually picking up these lymph nodes.
Okay, great. I was thinking of that distance from tumor question and how you could use it there. Alex, do you have follow-up questions?
Yeah, I had one that I'm maybe taking things a little bit sideways, but I know you'd mentioned a veterinary application as well. And I'm curious if you've developed a strategy for approaching the veterinary market, because particularly in Canada, but even in the United States, where there's a lot more government funding than a lot of people realize, it's a very different market dynamic. Where you don't have large hospitals as a central hub that can make substantial capital investments. So I'm wondering, because it's a very lucrative potential market, what your specific strategy is to go into the veterinary space, partly because it's somewhat less regulated than human medicine in some aspects. And so it can be an interesting place to test new concepts and see how they work.
Alex, I love that question. And that's something that we're hoping to build out with the IP and legal counsel, because we haven't explored that strategy in significant detail. But we are aware of these particular markets on how unregulated it may be in terms of bringing in a new system or technology like this into identifying lymph nodes in animal tissues. But that's something that we'd have to look into. I know the government of Canada had an innovation proposal for identifying lymph nodes in cows, for example. So there's a question of whether the similar type of application could be used for these in the context of other animal tissues. But again, it's something we'd have to work on and investigate in more detail. It is on our radar though.
Okay. Another question I had was about your return on investment. You mentioned that the $100,000 could be paid off in a year. Could you explain where the savings are going to come from for the hospital? Is it purely time, or is there something else?
Dr. Hamilton, that's a wonderful question. So before I sort of talk about it in a tiny bit more detail, what we're trying to do is develop an health economics of adoption model for our device. So there's a few ways this could be approached in terms of the cost savings element. One would be to really look into the current cost with manual dissection. So the existing methods, how much does that cost pathology labs? Potential cost savings developed by our device, and maybe other models we could explore. So the next slide that I'll be showing you incorporates models 1, 2, and 5 together. And this is essentially what I'm showing. Now, obviously, there's a lot of detail here, and I won't go into too much detail. I can follow up afterwards. But what we're trying to do is sort of categorize these different elements of our cost savings calculator. One would be trying to investigate sort of a revenue generating opportunity or a faster throughput method for pathology labs. And the other element would be in terms of material savings for pathology labs. So for the first category, again, I'll speak very high level. If our device can save pathology staff some time to concurrently work on other samples, we have an estimated amount of time that our device could save pathology staff every year. So what I mean by here is we're not replacing pathology staff. It would basically help free up some of the time that they can work on other samples concurrently. Mm-hmm. So here I have about 330 hours. And if we were to allocate about 1.5 hours for a new specimen coming in, then theoretically these labs can process an additional 220 samples. And this goes back to Lou's question, where breast cancer specimens are coming in nonstop, right? And when we factor in sort of the reimbursement code for each of these specimens, pathology labs would see a faster throughput, and they would see sort of a revenue generating opportunity with our device. For the second element, with the existing manual process, there's a lot of pathology waste being produced because oftentimes all the lymph nodes are not found properly. So pathology staff are submitting extra tissue regions for further processing. So what we calculated is with our device, if you can save certain number of cassettes, then this would be the hard material savings for pathology labs. And again, this is just a preliminary model that we've worked out, and we're validating with pathology administrators at the moment.
Okay, great. We're almost out of time. Are there any other burning questions, Lou or Alex?
No, it's great. Thanks very much for the presentation.
Thank you, Samik. Appreciate it.
Thank you
very
Samik,
much.
and thanks to your team as well. I know they're behind all of these slides as well. So thank you very much.
Yes, thank you so much, guys.
Thank you.